Good afternoon, dear students. Today we will continue to study a very important section of special microbiology devoted to anaerobic spore-forming microbes like Clostridia and the causative agent of zoonotic infection like anthrax. The phenomenon of anaerobiosis and its global significance in biology, physiology, and pathology. Louis Pasteur in 1861, on the example of butyric acid fermentation bacteria, proved the existence of microbes that require oxygen-free growing conditions and proposed the term of anaerobes or anaerobic bacteria. Anaerobic digestion of organic matter by microbes is called fermentation. The most well-known alcoholic, lactic, acetic, diuretic, pyronic types of fermentation. Classification of bacteria according to the type of respiration. All microbes, according to the type of respiration, can subdivide it into four groups. The first group, obligate or strict aerobic bacteria. As example of obligate strict aerobic bacteria, we can use causative agent of tuberculosis, Mycobacterium tuberculosis. The second group is obligate or strict anaerobic bacteria. For this group, we want bacteria like Clostridium tetani, causative agent of tetanus, Clostridium botulinum, causative agent of botulism, Clostridium septicum, Clostridium histolyticum, Clostridium perfringens. This is the causative agents of gas gangrene. The third group, according to the type of respiration, it's facultative anaerobic bacteria. As example, it's Escherichia coli, causative agent of Escherichiosis, and other members of family Enterobacteriaceae. And the last group of bacteria, according to the type of respiration, it's microaerophiles. As example, it's causative agents of brucellosis, brucella, and also Helicobacter and Campylobacter pylori. Obligate anaerobic bacteria can grow only in the absence of oxygen. From a biochemical point of view, obligate anaerobes are bacteria that do not use oxygen as ultimate electron acceptor. These bacteria obtain energy either by fermentation. The ultimate electron acceptors are organic compounds, or by anaerobic respiration. This way, electron acceptors are inorganic, oxygen-containing compounds like nitrites, sulfides, carbon dioxides. Obligate anaerobic bacteria do not contain oxidase, catalase, peroxidase, cytochromes, and some particular strict anaerobes also lack flavin enzymes. Flavin enzymes used for their biochemical identification. Obligatory anaerobic bacteria are cultured only in the absence of oxygen. Microaerophiles in conditions of its low partial pressure. In the presence of oxygen, obligate anaerobic bacteria die. And why is oxygen detrimental to anaerobic microbes? In the process of metabolism, in the presence of oxygen, all microbes form highly reactive forms of oxygen, like superoxide anion, hydrogen peroxidase, hydroxylated. If these peroxidase are not neutralized, the microbes experience oxygen stress and die. Neutralization of peroxide compounds in anaerobic and facultative anaerobic microbes occurs in two ways: enzymatic and non-enzymatic. Enzymatic method of inactivation of peroxidase compounds is carried out by enzymes. It's microbial superoxidase mutase, catalase, and peroxidase, and non-enzymatic neutralization of peroxidides is carried out mainly with the help of glutathione. In this slide you can see main anaerobic microbes which are medically important in microbiology. All these microbes are divided into spore-forming anaerobic microbes and non-spore-forming. Spore-forming, like example, you can see it's Clostridium. That about Clostridium tetani, Clostridium perfringens, Clostridium novae, Clostridium histolyticum, and other bacteria. And for non-spore-forming anaerobic bacteria, non-actinomycetes, Lactobacillus, Propionibacterium, Peptococcus, Peptostreptococcus, Bacteroides, Fusobacterium, Treponema, Borrelia, Veillonella, and many other bacteria. Main features of cultivation and isolation of pure culture of anaerobic bacteria. The main condition is to prevent contact of the material and the isolated culture with oxygen. To do this, certain rules are used for the collection of material, its transport, shown under anaerobic condition, on special nutrient medium, from which oxygen is removed by various methods. For example, by physical methods, we use boiling the culture medium, anaerobic culture apparatus, desiccator, and other chemical methods. It's chemical oxygen absorption and oxygen reduction, and different biochemical methods. For example, it's Fortner and Peretz method. This system is used for the anaerobic incubation of up to 12 petri dishes in anaerobic jar. This reagent contains components which create an oxygen-free anaerobic environment in the jar and simultaneously release carbon dioxide. The contained oxygen does not react with hydrogen to form water, but is chemically bound to the finally distributed iron powder in the reagent mixture. The nutrient media on which anaerobes are grown are similar in composition to universal media. They differ only in that they themselves must create anaerobic conditions. For this purpose, the culture media are poured in a high column. Liquid media are boiled to remove oxygen from them, and vaseline oil is laid on the surface to isolate from atmospheric oxygen. In addition, the composition of nutrient media includes various reductant substances that reduce the content of free oxygen in them. For the isolation of anaerobic bacteria from the pathological material, as well as for their accumulation, a liquid nutrient medium, ketotarotsi, is used. The nutrient medium contains hepatic extract with meat infusion broth or meat infusion bouillon. Pieces of boiled liver are preliminarily placed in the bottom of the test tube, and vaseline oil is laid on top after the medium has spilled. General properties of Clostridia. Clostridia is large rods, gram-positive, with rounded ends. Most species mobile in anaerobic conditions and has flagella. It's peritrachea. The spore in Clostridium diameter of spore greater than the diameter of vegetative cell. Clostridia live in large intestine of animals, and also Clostridia can be isolated from the human large intestine. The biochemical activity of Clostridia is generally quite high. Carbohydrates are fermented to form acid and Hes. I want to pay attention to the high biochemical activity of His gangrene was first described in 1562 by Ambroise Paré, who gave it the name of hospital infection. Most often, case gangrene is caused by the association of Clostridia with the accession of other microbes. Most cases of case gangrene are the results of the action of Clostridia, always Clostridia perfringens, and aerobic associative bacteria. The main clostridial associate on which depends the nature of the course and clinic of the disease is Clostridium novi, Clostridium septicum, Clostridium histolyticum, Clostridium sardelli, Clostridium ramosum, and other. Anaerobic infections may be caused by any one of the first four species mentioned above, but usually several members of the parasitomosis action in a particular combination are responsible for them. The less pathogenic and non-pathogenic species can't be responsible for anaerobic infection by themselves, but they cause tissue destruction, low with the oxidation reduction potential, and thus create favorable conditions for the growth of pathogenic species. The pathogen was discovered in 1892 by Welch and Natal. These Clostridia are most represented in the human intestine, 1,000 to 100,000 bacteria per 1 gram of feces. Features of morphology. Clostridium perfringens is gram-positive, rod-shaped bacteria. In contact to other Clostridia have no flagella. In the affected tissues have a massive capsule. The exudate smears are characterized by the absence of neutrophils. Spore formation is not intense. Spores are often located central or subterminally. By type of respiration, it is micro-air tolerant microbe. According to biological characteristics, Clostridium perfringens is divided into 6 types: A, B, C, D, E, and F. They produce 12 toxins and enzymes with different antigenic properties, which are denoted by the letters of the Greek alphabet: alpha, beta, gamma, delta, epsilon, kappa, lambda, mu, and others. For humans, pathogenic types A, C, and D, types B, C, D, and E mainly cause disease in farm animals. The basic type A, produces alpha toxin, glycitinase C, which has lethal, necrotic, neurotoxic, and hemolytic properties. Plays a major role in the pathogenesis of anaerobic gas infection. Enzymes of pathogenicity: collagenase, hyaluronidase, protease, DNase. These enzymes can destroy tissue biopolymers. Beta, epsilon, and iota toxins have lethal and necrotic properties. Gamma and eta toxins are less lethal. Delta toxin has a lethal effect and causes hemolysis of erythrocytes of cloven-footed animals, sheep, goats, pigs, cats. Theta toxin is an oxygen-labile hemolysin having antigenic similarity to streptolizine O. It causes death and cytosis. Kappa toxin is a collagenase. Lambda toxin is a protease and gelatinase. Mu toxin is hyaluronidase. And mu toxin is a DNase. Clostridium perfringens can be identified by growth on egg agar. On this medium, colonies of Clostridium perfringens are surrounded by an opalescent white precipitate that appears under the action of alpha toxin of Clostridium perfringens, its lecithinase C. Also on blood agar, Clostridia perfringens form colonies surrounded by a zone of complete or partial hemolysis. There are the following forms of anaerobic gas infection. Anaerobic infection of the soft tissues of the extremities and torso. Anaerobic brain infection. Postpartum or post-abortion anaerobic infection. Anaerobic infection of the abdominal cavity and peritoneum. And anaerobic infection of thoracic cavity. The last is anaerobic osteomyelitis. After entering of Clostridia perfringens the wound, under conditions of hypoxia, spores grow into vegetative cells. Clostridia multiply at the entrance gate and synthesize exotoxins and enzymes of aggression. In particular, lecithinase breaks down lecithin of cell membranes. Hyaluronidase and collagenase increase tissue permeability. The result of the reaction of toxins and exoenzymes is the necrosis of muscle and connective tissues, the development of edema, The accumulation of gases in tissues, and intoxication of the body. The conditions for the development of clostridial infection are traumatic tissue necrosis and the presence of anaerobic. The incubation period for Clostridial anaerobic infection is 1 to 3 days. The skin around the wound is pale with a bluish tinge. The edges of the wound is swollen. Visible muscles resemble boiled meat. When pressed, gas bubbles with unpleasant odors are released from the wound. On palpation, characteristic crepitus is noted. General intoxication of the body is expressed. The main cause of lethal inheritance is the degeneration of the parenchymal organs and the damage to the central nervous system by tissue degradation, byproducts, and microbial toxins. Gas a mixture of methane, hydrogen, carbon dioxide appears in the tissues. There is also hydrogen sulfate, ammonia, other gases with unpleasant odor. Gas bubbles create a characteristic repetition effect. This slide shows various forms of gas gangrene with limb involvement. Focuses of soft tissue necrosis, vesicles with serous consent, and purulent discharge from wounds are visible. A common complication of gas gangrene is limb amputation. Material for investigation of the Clostridial anaerobic infection is the affected and necrotic tissues taken from the wound of the bore with healthy tissue, exudate, pus, wound discharge, and blood. In laboratory diagnostics of Clostridial anaerobic infection, we use bacterioscopic, cultural, and biological methods of laboratory diagnostics. The bacterioscopic method includes microscopy of smears prepared from the test material and stain by gram and Bourigin's method for identification of bacterial capsule. The culture method involves inoculation of the test material into a liquid nutrient medium, for example, Kit-Tarotse medium, for the accumulation of bacteria, followed by the reinoculation of the culture on solid nutrient medium. The biological method is based on the reaction of neutralization of toxins with specific antitoxic serum, followed by infection of white mice. Also, we can use accelerated bacteriological diagnostics. On the slide, you can see coagulation of milk, ruptures of glucose agar column, blackening of the iron sulfide medium of Wilson-Blair. Treatment. The main attention is paid to the surgical treatment of the wound, removal of necrotic tissues, ensuring of outflow of the separated wound, drainage, and the flow of oxygen, the use of dressing with hydrogen peroxide solution. To provide tissues with oxygen, hyperbaric oxygenation is used to create an increased oxygen concentration in the blood. An oxygen concentration of 3% creates a bacteriostatic effect. In oxygen concentration of 6%, bactericidal effect. Neutralization of Clostridium toxins is carried out using The causative agent of tetanus was discovered almost simultaneously by Monastyrsky in 1883 and Nikolaev in 1884. Kitazata was first isolated it in pure culture. Tetanus is a severe disease mediated by the neurotoxic effect of bacterial exotoxin, tetanus plasmin. The leading manifestation is a convulsive syndrome, including muscle contractions, tetanus, and prolonged muscle tension. Epistatonus is considered a characteristic manifestation of tetanus. Clostridium tetani is gram-positive bacteria, informants rod-shaped bacteria with rounded ends from 4 to 8 microns long and 0.3 to 0.8 microns thick. In young cultures, it sometimes form filamentous cells. Allocated singly or in chains, these bacteria, mobile, contain 20 or more flagella located along cell periphery, but in old cultures 30 days and more, immobile forms. These bacteria obligate or strict anaerobes. These bacteria are highly sensitive to oxygen, and Clostridium tetani like enzymes like cytochromes, cytochrome oxidase, peroxidase, and catalase. Under strictly anaerobic conditions, the pathogen grows slowly and forms thin transparent colonies with smooth or rough edges, and growth of colonies is characteristic. First, a mesh form of many colonies appear on the surface of the medium. Growth in the form of transparent or grayish yellow rough and smooth colonies. When inoculated with the column in semi-liquid agar, after 24 to 48 hours, forms colonies in the form of lentils, air forms, or fluffs with a dense brown center, S forms. Spores are round, less often oval, arcuated terminal. Their diameter is in 2 to 3 times greater than the thickness of bacteria, as a result of which the sporangium has the shape of a tennis racket or a drumstick. They are distinguished by high resistance to chemical and physical influences. In particular, they survive for 8 to 10 hours in 1% mercury chloride solution and 5% phenol solution, and also withstand boiling for 0.5 to 1 hours. Spore formation starts on days 2 to 3. On 4 to 6 days of growth in the liquid medium, vegetative cells are destroyed and almost only spores remain in the medium. Clostridium tetani produces at least two distinct toxins, ecamolysine, tetanolysine, and powerful neurotoxin tetanospasmin. There are two antigenically and pharmacologically distinct, and their production is mutually independent. Tetanolysine is a heat-label, oxygen-label hemolysin, antigenically related to the oxygen-label hemolysins produced by Clostridium perfringens and Clostridium novae. This lysis is active against erythrocytes of several animal species, especially rabbit and horse. Its pathogenic role is unknown. It may act as a leukotoxin. Tetanospasmin is the toxin responsible for the clinical manifestation of tetanus. It is oxygen-stable but relatively heat-labile, being inactivated at 65 degrees centigrade in 5 minutes. It is plasmid-coated. It gets toxoided spontaneously or in the presence of low concentrations of formaldehyde. It is a good antigen and it's specifically neutralized by the antitoxin. The toxin has been crystallized. It is a simple protein composed of a single polypeptide chain. On release from the bacterium, it is autolyzed to form a heterodimer consisting of a heavy chain and a line chain joined by a disulfide bond. Clostridium tetani has little invasive power. The spore implanted in the wound can germinate and multiply only if the conditions are favorable. The toxin produced locally is absorbed by the motor nerve ends and transported to the central nervous system along the axis cylinders of the peripheral nerves. The toxin is specifically and avidly fixed by gangliosides of the gray matter of the nervous tissue. Tetanospasmin resembles strychnine in its effects. The tetanus toxin specifically blocks synaptic inhibition in the spinal cord, presumably at inhibitory terminal that use glycine as a neurotransmitter. The toxin acts presynaptically, unlike strychnine, which acts postsynaptically. The toxicity of tetanospasmin is influenced by the route by which it is administered. Given orally, it is destroyed by the digestive enzymes and is without effect. Subcutaneous, intramuscular, and intranervous injection are equally effective. Intranerval injection are more lethal, and injection directly into the certain nervous system were much more tolerable. The route of administration also modifies the clinical picture. Tetanus results from the infection of a wound or surface with the tetanus bacterium, or more rarely from the parenteral injection of contaminated substances, and is characterized by tonic muscular spasms, usually commencing at the site of infection, and in all by the mildest cases becoming generalized, involving the whole of the somatic muscular system. Most frequently, the disease follows injury, which may even be too trivial to be noticed. Puncture wounds are particular uh, as a favor the growth of the anaerobic bacterium. Rarely it may follow surgical operation, usually due to sepsis and asepsis. Sometimes the disease may be due to local suppuration, such as otitis media, pathogenic tetanus. Tetanus is an important complication of septic abortion. It may be caused by unhygienic practices, such as application of cotton of the umbilical stump, of rituals such airborne or circumcision. Tetanus may also be caused by non-sterile injections, where contamination of a wound with tetanus spores does not invariably lead to tetanus. Unless anaerobic conditions are available, the spores remain dormant. Destruction and necrosis of tissues, lack of drainage in the area, presence of extranervous matters, especially of soil, infection with other bacteria, all cause the development of tetanus. Spores may sometimes remain dormant at the site of lodgement for a long time and suddenly erupt into activity either at the original site or at the other site when conditions become favorable. The incubation period is variable, from 2 days to several weeks, but is commonly 6 to 12 days. This is influenced by several factors such as the site and nature of the wound, the dose and toxicity of the contaminating organism, and the immune status of the patient. The incubation period of prognostic significance, the prognosis being great when it is short. Of similar significance is the interval between the first symptom of the disease, usually trismus, and the onset of spasm, period of onset. Tetanus was a serious disease with a high rate of mortality, from 80 to 90% before specific treatment became available. Even with proper treatment, The case fatality rate varies from 15 to 50%. Tetanus neonatal and uterine tetanus have very high fatality rates from 70 to 100%, while autogenic tetanus is much less fatal. Tetanus is more common in the developing countries, where the climate is warm, and in rural areas, where the soil is fertile and highly cultivated, where human and animals population are substantial and live in close association, and where unhygienic practices are common and medical facilities poor. The diagnosis of tetanus should always be made on clinical grounds. Laboratory tests only help in confirmation. Not infrequently, it may be not possible to establish laboratory diagnosis at all. Laboratory diagnosis may be made by demonstration of Clostridium tetani by microscopy, culture, or by animal inoculation. Microscopy is unreliable, and the demonstration of the typical drumstick bacteria in wound is in itself is not diagnosis of tetanus. The bacteria may be present in some wounds without tetanus development. It may not also be possible to distinguish by microscopy between Clostridium tetani and morphologically similar bacteria such as Clostridium tetanomophum and Clostridium sphenoides. Diagnosis by culture is more dependable. Isolation is more likely from exuded bits of tissue from the necrotic depths of wounds that form wound swab. The material is inoculated on one half on a blood agar plate. Clostridium tetani produces swimming holes, which may be detected into the opposite half of the plate after 1 to 2 days incubation anaerobically. The material is also inoculated into 3 tubes of cooked meatballs, one of which is heated to 80 degrees centigrade for 15 minutes, the second for 5 minutes, and the third left unheated. The purpose of heating for different periods is to kill vegetative bacteria while leaving undamaged tetanus spores, which very widely is heat resistant. The cooked meat tubes are incubated at 37 degrees centigrade and subcultured on one half of blood agar plates daily for up to 4 days. Clostridium tetani may be isolated in pure culture by subculturing from the swimming edge of the growth. Toxigenicity is best tested in animals. At 2 to 4 days, all cooked meat culture, 0.2 milliliters, is inoculated into the root or the tail of a mouse. The second mouse that has received the tetanus antitoxin in hour earlier serves as a control. Symptoms develop in the test animal in 12 to 24 hours, beginning with stiffness in the tail. Rigidity proceeds to the leg on the inoculated side, the opposite leg, trunk, and forelimbs in that other. The animal dies within 2 days but may be killed earlier as the appearance of ascending tetanus is diagnostic. Tetanus patients should be treated in hospitals in special units. The reason for isolation then is to protect them from noise and light which may provoke their convulsions. Tetanus patients hardly have infections, and person-to-person transmission does not occur at all. Passive immunization is by injection of tetanus antitoxin, anti-tetanus serum from hyperimmune horses was the preparation originally used. Active immunization is not only the most effective method of prophylaxis, but also the only means whereby tetanus, following innatized injuries, can be prevented. This is achieved by spread injections of formal toxoid called DTP, diphtheria tetanus toxoid, diphtheria tetanus pertussis toxoid. Clostridium botulinum causes botulism, a severe form of food poisoning. The name botulism is derived from sausage, from Latin word botulus, sausage, an article of food used to be associated with this type of food poisoning. Clostridium botulinum was the first isolated by van Hermingen in 1896 from a piece of ham that caused an outbreak of botulism. The bacteria is widely distributed saprophyte, occurring in virgin soil, vegetables, hay, silage, animal manure, and sea mud. Vegetative cells are rods with rounded ends measuring from 4 to 8 and 0.6 to 0.8 nanometers. This bacteria is mobile, has flagella, peritrichous flagella. Under unfavorable conditions, endospores are formed. The spore is located terminal or subterminal. This bacteria is strict anaerobic bacteria. Young cultures are stained gram-positive, and after 4 or 5 days, all cultures stained gram-negative. Optimum pH for growth of this bacteria is 7.3–7.6. For spore fermentation, 6 to 7.2. On blood agar with glucose, very small grayish or cloudy, yellowish lenticular colonies are formed. Zones of hemolysis of various weights are formed around the colony. On liver agar, polymorphic stellate colonies are formed on gelatin, grayish, surrounded by a zone of liquefied gelatin. Dissociates can be found on the agar column. Air form are in the form of lenticular grains. S form are fluffy. They grow well in liquid media, usually medium of kit tarotse, provided that oxygen is removed from the medium by boiling for 15-20 minutes with rapid cooling. Eight types of Clostridium botulinum can be identified. Type A, B, C1, C2, D, E, F, and G. The toxins produced by the different types are identical in their pharmacological activity, but are neutralized only by of gamma-logous anticillin. An exception is C2 toxin, which is cytotoxin that causes increased vascular permeability, while all other are the neurotoxins. Until recently, it was believed that any identical strain produced only one type of toxin, but a strain has been isolated which produced both A and F toxins. The toxins are placed in three groups because of some antigenic relationships. Clostridium botulinum produces a powerful exotoxin that is responsible for its pathogenicity. The toxin differs from other exotoxins in that it is not released during the life of the organism. It is produced intracellularly and appeared in the medium only on death and autolysis of the cell. It is believed to synthesize initially as a non-toxic protoxin or progenitor toxin. Trypsin and other proteolytic enzymes activate progenitor toxin to active toxin. Toxin has been isolated as a pure crystalline, which is probably the most toxic substance known. The lethal dose for human beings is probably 1 to 2 nanograms. It is a neurotoxin, acts slowly, taking several hours to kill. The toxin is relatively stable, being inactivated only after 30 to 40 minutes in 80 degrees centigrade and at 10 minutes at 100 degrees centigrade. Food suspecting to be contaminated with botulotoxin can be rendered completely safe by cooking or boiling for 20 minutes. It is resists digestion and is absorbed through the small intestines in active form. It acts by blocking the producer release of acetylcholine at the synapses of and neuromuscular junctions. The onset is marked of diplopia, dysphagia, and these are clear due to cranial environment. Asymmetric descending paralysis is a characteristic pattern ending in death by respiratory paralysis. A small quantity of Clostridium botulinum type A toxin injected in the muscle selectively weakens in the blocking the release of acetylcholine at the neuromuscular junction. Muscles so inject atopy by recovery in 2 or 4 months is a new terminal axon approach form and restore transmission. Intramuscular injection of the toxin, the first used to treat Strabismus, is now recognized as a safe and effective symptomatic therapy for many neuromuscular diseases. The botulotoxin Clostridium botulinum is non-invasive and virtually non-infectious. Its pathogenicity is due to the action of its toxin, the manifestation of which are collectively called botulism. Botulism is of three types: food-borne botulism, wound botulism, and infant botulism. Food-borne botulism is due to ingestion of preformed toxin. The types of the bacterium and the nature of the food responsible vary in different regions. Human disease is usually caused by type A, B, and E, and very rarely type F. Type C and D are usually associated with outbreaks in cattle and wild fowl. Type G has been associated with sudden death in the few patients. The source of botulism is usually preserved food, meat and meat products in Europe, canned vegetables in America, and fish in Japan. Type E is associated with fish and other seafood. Proteolytic variants of Clostridium botulinum can digest food, which then appears spoiled. The cans are often inflated and show bubbles on opening. Non-proteolytic variants leave food unchanged. Symptoms begin usually from 12 to 36 hours after ingestion of food. Vomiting, thirst, conspiration, ocular paresis, difficulty of swallowing, speaking and breathing constitute the common features. This is due to respiratory failure and occurs from 1 to 7 days after onset. Case fatality varies from 25 to 70%. Wound botulism is a very rare condition resulting from wound infection with Clostridium botulinum. Toxin is produced at the site of infection and is absorbed. The symptoms are those of foodborne botulism except for the gastrointestinal components, which are absent. Type A has been responsible for most of the cases situated. Infant botulism was recognized as a clinical entity in 1976. This is a toxico-infection. Clostridium botulinum spores are ingested in food, get established in the gut, and there produce the toxin. Cases occur in infants below 6 months. Older children and adults are not susceptible. The manifestations are constipation after a period of normal development, poor feeding, lethargy, weakness, poor oral secretory, lack of alter cry, weakness, and loss of head control. Patients excrete toxin and spores in their faces. Toxin is not generally demonstrable in blood. Management consists of supportive care and assisted feeding. Antitoxins and antibiotics are not indicated. Degrees of severity vary from very mild. Mechanical ventilation must be maintained at foodborne botulism. Immediate intravenous use of polyvalent antitoxin against serotype A, B, and E is essential. However, tests must be conducted to determine hypersensitivity to the antitoxin and the potential necessary for densitization prior to its administration. Infant botulism requires only supportive therapy. Prophylaxis. As most cases of botulism, following consumption of inadequately canned or preserved food, control can be achieved by proper canning and preservation. When an outbreak occurs, a prophylactic dose of antitoxin should be given intramuscularly to all who consumed the article of food. Active immunization has been shown to be effective. If immunization is needed, as in laboratory works exposed to the risk to injection of aluminum sulfate adsortoxoid may be given at an interval of 10 weeks, followed by the booster a year later. Antitoxin may be tried for treatment. Polyvalent antiserum to type A, B, and E may be administered as soon as a clinical diagnosis is made. abortive therapy with maintenance of respiration of the equal of greater importance. In addition, a combination of guanine tetrachloride and germine monoacetate may be administered bearing in mind their potential toxicity and side effects. Diagnosis may be confirmed by demonstration of the bacterium or the toxin and food in food or feces. Gram-positive spore-forming bacteria may be demonstrable in smears made from the food. Clostridium botulinum may be isolated from the food or the patient feces. The food is macerated in sterile sodium chloride solution and the filtrate inoculated into the mice or guinea pigs intraperitoneally. Control animals protected by polyvalent antitoxin remain healthy. Typing is done by passive protection with type-specific antitoxin. The toxin may occasionally be demonstrable in the patient's blood or in the liver post-mortem. A retrospective diagnosis may be made by detection of antitoxin in the patient's serum, but it may not be detectable in all cases. The next slide shows the susceptibility of animals to botulinum toxin. The family Bacillacea comprises a highly diverse group of endospore-forming bacteria. The genus Bacillus includes 51 described species and many additional species of uncertain taxonomic strains. Classification of pathogenic and conditionally pathogenic bacillus. Family Bacillacea. Genus Bacillus. Pathogenic species Bacillus anthracis. And conditionally pathogenic species it's Bacillus cereus, Bacillus subtilis, Bacillus mesentericus, Bacillus megatherium, and others. The genus Bacillus consists of aerobic bacilli forming heat-resistant spores. They are gram-positive but tend to be decolorized easily so as to appear gram-negative or gram-variable. They are generally motile with peritrichous flagella, the Andrus bacillus being a notable exception. Members of these groups exhibit great diversity in their properties. The genus includes psychrophilic, mesophilic, and thermophilic species. The maximum temperatures of vegetative growth ranging from about 25 degrees centigrade to above 75 degrees centigrade and the minimums from 5 to 45 degrees centigrade. The anthrax bacillus is one of the largest of pathogenic bacteria. In tissues, it is found singly, in pairs, or in short chains, the entire chain being surrounded by a capsule. The capsule is polypeptide in nature, being composed of a polymer of D-glutamic acid. Capsules are not formed under ordinary conditions of culture, but only if media contain added bicarbonate or incubated in 10 to 25% of carbon dioxide. If grown in media containing serum, albumin, capsule formation may occur in the absence of carbon dioxide. In cultures, the bacillus are arranged end-to-end in long chains. Spores are formed in culture or in the soil, but never in the animal body during life. Spirulation occurs under unfavorable conditions for growth, is encouraged by distilled water, 2% of sodium chloride solution, or grows in oxalated agar. Spores are formed only in the presence of oxygen, and spirulation is inhibited by anaerobic conditions and by calcium chloride. Spores are central, elliptical, or oval in shape, and are of the same width as the bacterial body. The anthrax bacillus is gram-positive and non-acid-fast bacteria. The spores do not stain by ordinary methods, but can be stained differentially by special techniques. It is an aerobe and facultative anaerobe with temperature range for growth of 12 to 45 degrees centigrade. Optimum temperature is 35 to 37 degrees centigrade. The optimum temperature for spirulation is 25 to 30 degrees centigrade. Good growth occurs on ordinary medium. On agar plates, irregular round colonies are formed 2 to 3 millimeters in diameter, raised, dull, opaque, grayish-white, with frosted glass appearance. Under the low power of microscope, the age of the colony is composed of long chains of bacillus resembling a lock of matted hair. This is called the Medusa head appearance. Virulent capsulated strains from reef cultures bite a virulent or attenuated strains form smooth colonies. On gelatin stub culture, characteristic inverted hair tree. On blood agar, the colonies are non-hemolytic. In broth, growth occurs as follicular deposit with little or no turbidity. When Bacillus anthracis is grown on the surface of a solid medium containing penicillin, in 3 to 6 hours the cells become large, spherical, and occur in change on the surface of the agar, resembling a string of pearls. The strings of pearls reaction differentiates Bacillus anthracis from Bacillus cereus and other aerobic spore-forming bacteria. Another useful test to differentiate Bacillus anthracis from Bacillus cereus is the form's susceptibility to gamma phages. Anthrax toxin was identified by the find that the injection of sterile plasma of guinea pig dying of anthrax into healthy guinea pig killed them and their death could be prevented by immune serum. The toxin is a complex of three fractions. The first is the edema factor or EF factor. The second is the protective antigen factor, PA factor. And the first is the lethal factor or LF factor. These factors are not toxic individually, but the whole complex produces local edema and generalized shock. The three factors have been characterized and cloned. Protective antigen factor is the fraction which binds to the receptors on the target cell surface and in turn provides attachment sites of EF edema factor or LF lethal factor, facilitating their entry into the cell. Antibody to protective factor is protective because it blocks the first step in toxin activity, it is binding to target cell. EF factor is an adenylyl cyclase which is activated only inside the target cell, leading to intracellular accumulation of cyclic adenosine monophosphate. This is believed to be responsible for the edema and other biological effects of the toxin. Entry of the lethal factor into the target cell causes cell death, but the mechanism of action is not known. Loss of the plasmid, which encodes the anthrax toxin, renders the strain virulent. This is believed to have been the basis for the original anthrax vaccine developed by Pasteur. Large epidemics of anthrax have been reported from Russia and Zimbabwe during 1978-1980 years. Human anthrax is contracted from animals directly or indirectly. The disease may be cutaneous, pulmonary, or intestinal, and all types leading to fatal septicemia. Cutaneous anthrax follows entry of the infection through the skin. The face, neck, hands, arms, and back are the usual sites. The lesion starts as a papule from 1 to 3 days after infection and becomes vesicular, containing fluid which may be clear or blood-stained. The whole area is congested and edematous, and several satellite lesions filled with serum or yellow fluid are arranged around a central necrotic lesions, which is covered by a black scar. The name anthrax, which means fall, comes from the black color of the scar. The lesion is called a malignant pustule. The disease used to be common on dog walkers carrying loads, hides, and skins of their bare back, and hence was known as the hide porter's disease. Cutaneous anthrax generally resolves spontaneously, but 10 to 20% of untreated patients may develop fetal septicemia or meningitis. Pulmonary anthrax is called the wool-sorter's disease because it used to be common in workers in wool factories due to inhalation of dust from infection wool. This is hemorrhagic pneumonia with a high fatality rate. Hemorrhagic meningitis may occur as a complication. Intestinal anthrax is rare and occurs mainly in primitive communities with the carcasses of animals dying of anthrax. Enteritis with bloody diarrhea occurs with high case fatality. Human anthrax may be industrial or non-industrial or agricultural. Non-industrial anthrax is often an occupational disease in those who associate with animals, such as veterinarians and farmers. Anthrax may be diagnosed by microscopic culture, animal inoculation, and serological demonstration of the anthrax antigen in infected tissues. The immunofluorescence technique can be adopted for identifying of Bacillus anthracis in smears. Acute and convalescent phase sera should be obtained since antibodies to the organism can be demonstrated by gel diffusion, complement fixation test, and the LISA technique. The type of test to be employed depends on the nature of the material available. When an animal is suspected to have died of anthrax, autopsy is not permissible as the split blood will lead to contamination of the soil. An ear may be cut off from the carcass and sent to laboratory. Alternately, swab soaked in blood or several blood smears may be sent. The demonstration of gram-positive bacillus with the morphology of anthrax bacilli in positive MUC5 reaction will enable the presumptive diagnosis to be made. Isolation of the bacillus is easy of cross contamination has not occurred. The anthrax bacillus can often be isolated from contaminated tissues by applying them over the shaven skin of the guinea pig. It is able to penetrate through minute abrasion and produce fatal infection. If the sample received is putrid, though that viable bacillus are unlikely, diagnosis may be established by a Scully thermoprecipitation test by demonstration of the anthrax antigen in tissue extracts. Specific prevention of anthrax. The first live vaccine was created by Louis Pasteur in 1882. In Russia, a live